So Andrew is currently in his 6th year of a combined MD PhD in pharmacology under the supervisions of Dr. Stefan Burk and Ferenc Gragazin. His research focused on how perinatal iron deficiency impacts cardiovascular development and function, is supported by a CIHR Vanier, very impressive, and an Alberta Innovates graduate studentship. So welcome, Andrew. Take it away.
Well, thank you to you and the committee for giving me the opportunity to present today. As you mentioned, I'm going to be talking about iron deficiency and how it impacts nephrogenesis and how it impacts vitamin A metabolism as well. So as we just heard, anemia is a prevalent occurrence in pregnancy, and it's a major global health concern. As you can see here, the prevalence of anemia is quite striking globally. This is data from 2011 from the World Health Organization. You can see that most nations have a prevalence of approximately 20 to 40% throughout pregnancy, whereas other areas such as developing nations or malaria endemic nations have a much higher percentage, with rates of anemia exceeding that 60% level. So globally, the leading cause of anemia during pregnancy is iron deficiency, and it actually contributes to a majority of those cases. And this is because we know that iron demands during pregnancy greatly exceed the normal requirements. So you can see, especially in the 2nd and 3rd trimester, we have a huge increase in the total iron demands per day, and that's largely due to red cell volume expansion and the iron demands of the fetus itself. Now, in the short term, it's well established that this has quite severe negative health consequences for the offspring, and these include preterm birth, intrauterine growth restriction, and perinatal death. However, the long-term consequences of this have been underappreciated for quite a long time. There is evidence that iron deficiency does lead to neurologic deficits in developing offspring, but more and more now, and it's the focus of our lab, we're seeing evidence for cardiovascular dysfunction as well. So we've actually shown in our model things like hypertension and renal injury occurring in the adult offspring that were born iron deficient, and these tend to be much more severe in the male offspring versus females. And one of the reasons why we think we're seeing this hypertension and renal injury phenotype is actually due to an inborn reduction in the number of nephrons that these offspring have. So the number of nephrons we have is set at birth or shortly thereafter, depending on the species we're talking about. And what you have then is what you're stuck with for the rest of your life. So what I have here on the slide is actually a time lapse of a developing mouse kidney. And the green there is labeled for the ureteric bud. So the ureteric bud is sort of one of the primitive structures of the kidney that will continue to branch much like a tree throughout development. So you can imagine that if we reduce the amount of branching, we're going to reduce the amount of nephrons that we're seeing. And that's because the nephrons are going to form around the ureteric buds, or the sort of leaf-like features that you can see here on those images. Now, we also wanted to look at vitamin A in our model, and this is because vitamin A status is actually one of the tightest, most tightly associated metabolic or endocrinologic features that we know of that associates with glomeruli. So you can see from this paper from 1998 that vitamin A deficiency, as denoted by plasma retinol levels, very tightly correlates with the overall number of glomeruli that these offspring are born with. We also know that iron deficiency does impact vitamin A metabolism, so we thought that this would be a good rationale for exploring this in our own model. So just to give everybody a very quick primer on vitamin A metabolism. So we're going to be talking about how retinol comes into the kidney, particularly taken up by interstitial cells, and it'll be converted to retinaldehyde. The retinaldehyde will then undergo transformation to retinoic acid, which is the active metabolite of vitamin A. And that occurs through retinaldehyde dehydrogenase, which is a rate-limiting enzyme for this process. After that, the retinoic acid has a couple of different couple of different pathways it can undergo. So first, it could be degraded by CYP enzymes, most notably CYP26B1, that's the major isoform. Or it could actually be transported to the ureteric bud cells, where it will then bind to the retinoic acid receptor. And that induces transcription of the receptor tyrosine kinase known as RET. Now, RET is bound by glial-derived neurotrophic factor, or GDNF, and that induces a signaling cascade, which ultimately results in transcription of factors that contribute to this ureteric bud branching. And one of the notable factors that we're going to be talking about in that pathway is beta-catenin. So our objective in this study was to determine whether or not perturbations in renal vitamin A metabolism and signaling are associated with reduced nephron endowment in perinatal iron deficient offspring. So to give you a quick primer on our model, we take a bunch of Sprague Dawley dams and we randomize them either to a control iron replete diet, which has 37 milligrams per kilogram iron, or we place them on a low iron diet, which is 3 milligrams per kilogram. And we'll have those rats on the diet 2 weeks prior to pregnancy. After confirming pregnancy, we'll then take that iron restricted group and we'll place them on a moderately low iron diet, and that's just to ensure viability of the offspring. After giving birth, all of the rats, the offspring and dams, will be placed on that iron replete diet to allow replenishment of those iron stores over the course of the weaning and post-weaning environment. Now we're going to be looking at data from 2 different time points today, the first being postnatal day 1, or within 24 hours of birth, and then as well at day 28, which is a week after weaning, and that's a time at which the iron status of the offspring has normalized. Some of the methods we used include HPLC to look at vitamin A status, RT-PCR to look at expression of a lot of the signaling pathway components, and then we assessed nephron endowment using acid maceration. So to characterize the degree of anemia that we're seeing in our model, you can see maternal hemoglobin levels at the outset of gestation are reduced in our iron deficient group, which is the yellow bars throughout the presentation. You can also see that there is that hemodilution effect throughout the course of gestation in both the iron deficient and control groups. Interestingly as well, we assessed whether or not there were reductions in retinol levels during pregnancy, and we did note that iron deficient mothers had reduced circulating retinol levels at the end of gestation. When we look at the offspring, we can see that both male and female offspring exhibit approximately 50% reductions in hemoglobin. And we also see that male and female offspring exhibit about 25% reductions in body weight. You may also see that the male offspring tend to be larger than their female counterparts, irrespective of which perinatal treatment they belong to. So I'm going to jump forward now to the postnatal day 28 time point. And this graph here shows the number of glomeruli per offspring kidney. And what you can see is that iron deficiency only in the male offspring contributes to a reduction in the number of glomeruli each kidney has. The panel on the left shows how we actually assess this, and that's through the acid maceration technique, where the kidneys are essentially digested in concentrated acid, and what we're left with are those glomeruli, which are the little brown spheres there that are denoted by the arrows, as well as other cellular debris such as tubules and other components of the kidney. So we actually count those on a grid, and it's comparable to stereologic methods for determining nephron endowment. So with this in mind, knowing that we are seeing that reduction in nephrons, and that's particularly in the male offspring, now let's jump back to the postnatal day 1 time point and look at some of the mechanisms. So if you recall the schematic I already showed you with the retinoid levels, first let's assess whether or not the retinol and retinol esters coming into the kidney are actually in fact reduced. So retinol is more of a short-term storage. And as you can see, there's a reduction in the retinol levels due to iron deficiency, and that's most prominent in those male offspring. Whereas when we look at retinol esters, which are more of a long-term storage, we can see that there's a sex effect where females overall have a higher level of retinol esters, albeit we do see a significant reduction in the iron deficient females when compared to the control females. So already we can see that there are reduced levels of retinoids coming into these kidneys, although the etiology is a little different in males and females. It's also worth noting that we did assess both offspring liver and circulating plasma retinoid levels, and those weren't different. So looking at the signaling pathway now, starting with the retinaldehyde dehydrogenase, we can see that there's an iron deficiency and interaction effect, where iron deficiency increases expression predominantly in those males, and females, we don't see an iron deficiency effect at all. Similarly, with cytochrome P450 isoform 26B1, we can see that iron deficiency increases expression, again, most predominantly in that male perinatal iron deficient group. But again, we're not seeing that robust increase in females. Moving forward to the retinoic acid receptor alpha, which is a primary isoform of that receptor, we can see again an iron deficiency effect as well as an interaction effect, where again, this is predominantly occurring in that male perinatal iron deficient group where we're seeing that robust increase. And again, females compared to their control are not increasing. When we look at the transcriptional target of that retinoic acid receptor complex, we can see that the overall expression of RET is not changed due to either iron deficiency or sex. However, when we look at the ligand for RET, we can again see an iron deficiency effect and sex effect where there's a robust increase in expression due to iron deficiency in males, although we are seeing an increase in the females as well, albeit that's not significant with the post hoc test. As well, you'll note that the females overall have higher expression than the males. Finally, with beta-catenin, which is one of those downstream signaling components, we can see that iron deficiency again increases expression in males, whereas it doesn't appear to change the expression in females. So taken together, we can see that iron deficiency results in reduced kidney retinoid levels in offspring on postnatal day 1. And we also see that the male offspring exhibit a reduction in nephron endowment, which is associated with sex-specific alterations in the expression of retinoic acid signaling pathway components. So with this in mind, we think that the dysregulation of the vitamin A pathway could actually be underlying a lot of the consequences that we're seeing long-term with perinatal iron deficiency. And with that, I'd like to thank everyone for listening, our lab members, my supervisors, Dr. Bork and Dr. Gregason, as well as Dr. Clugston from the Department of Physiology for giving us so much help with the vitamin A project, as well as his lab tech, Sam Kinney, for running that HPLC for us. And with that, thank you, and I'll take any questions.